Good morning, everyone. Myself, Ganesh Kumar Verma. I am from Biochemistry Department, All India Institute of Medical Sciences, Rishikesh, India. So today I am going to present my research project. The title of my research project is the carcinogenic effect of heavy metals and its association with epigenetic changes in urothelial bladder cancer patients. Our study is based on the urothelial bladder cancer patients, how it happens in the presence of heavy metals, and what is the role of heavy metals in the cause of urinary bladder cancer through epigenetic changes. Now let's come to the background of this study. According to the Global Cancer Observatory, it has been reported that the urinary bladder cancer is the 17th most common malignancy present in India, and the bladder cancer represents significantly health problem due to its potential morbidity and mortality. Nowadays, urothelial bladder cancer is commonly associated with industrialized regions, and the most accountable risk factor for the cause of this urinary bladder cancer have been tobacco smoke along with some occupational chemical exposures, including heavy metals and some other pollutants. A massive increase in human exposure to heavy metals have been noticed due to industrial activities since last century, and this is an emerging understanding of carcinogenic effects of certain heavy metals is now being considered as a causative agents for urothelial bladder cancer. However, till date, there is no study has been reported for the cause of this. There is no study has been reported that the carcinogenic role of certain heavy metals in the etiology of urothelial bladder cancer has not yet been studied comprehensively or mechanistically. So this is the lacuna of this study or the research gap of this study. So based on this lacuna, we hypothesized that different kinds of heavy metals is ingested. After ingestion of this heavy metal from the food and water, it will mixed up in the blood, and from this blood, it will transport to the different parts of the tissue or organ, and it can also, this heavy metal will move to or will transport to the urine, where it will be excreted from the urine. There is the 4 layer of this bladder wall, bladder lining, transitional cell, some mucosal layer, muscle layer, and fat layer. So it has been reported that the tumor are present in a transitional cell layer. So in this tumor, we have to check whether the accumulation of this heavy metal present or not, and after accumulation, this heavy metal is involved in the epigenetic modification like 5-methylcytosine present in the CpG island of some tumor suppressor genes. So we have to check the level of this heavy metal concentration in the tumor region, and we have to check whether this heavy metal is involved in the epigenetic modifications or not. So our hypothesis can be heavy metals is involved. Heavy metal, is there any correlation between the heavy metal concentration and global DNA methylation or epigenetic modification? We have to check what kind of correlation is present in the concentration of heavy metal present in the tissue, tumor tissue, and the epigenetic modification like 5-methyl cytosine percentage. So based on this hypothesis, our present aim is to investigate the carcinogenic role of heavy metals through epigenetic changes in urothelial bladder cancer. Based on this aim, our objectives can be, first objective can be, our first objective was to assess the heavy metal concentration in blood, urine, and tissue samples from the cancer patient and healthy control. In the second objective was the assessment of global DNA methylation, which are present in the percentage of 5-methyl cytosine in tumor and adjacent healthy tissue. We have to assess the level of 5-methyl cytosine percentage, which are present in the global DNA, tumor tissue and adjacent healthy tissue. Now, what kind of methodology we have to use to prove this study that whether our heavy metal is involved in the cause of urinary bladder cancer or not? Whether this heavy metal is involved in the cause of heavy urinary bladder cancer through induce of epigenetic changes in urothelial bladder cancer patients. So how will I prove this study? So for to prove this study, we have recruited 50 patients, 50 urinary bladder cancer patients, and 50 healthy controls were recruited. Heavy metals were quantified using a technique known as inductively coupled plasma mass spectrometry in blood, urine, and tissues of those collected samples. In the next step, the global DNA methylation level was assessed by a global DNA methylation ELISA kit in cancer tissue as well as their adjacent healthy tissue, which were obtained from patients. After doing this, we will perform the statistical analysis. We have performed the statistical analysis by using the software Stata. And for graphical representation, we have used GraphPad Prism. For non-parametric test, we have used the Man-Whitney U test for the comparison of two groups. And our data was not in normal distribution, so we have used the median and interquartile range for to show the data or for the representation of the data. For correlation purpose, we have used here Spearman rank correlation for to determine the heavy metal levels in cancer tissue and global DNA methylation. Because our data was not in normal distribution, so here we have used Spearman rank correlation for to show the correlation between the heavy metal concentration and the percentage of 5-methyl cytosine. If the statistical significance p value is less than 0.05, then we can say that there is the statistical significance. The baseline parameters, here we have used 5 types of baseline parameters like age. We have found that mean age was 56 in cancer patient, whereas in the healthy control, the mean age was 31. The gender that we have found was 94% male in the case of cancer, whereas in the case of healthy controls, the male percentage was 66. So here we can say that cancer cases was more and more in the case of male because of this habit. smoking and alcohol. Smoking percentage was 37% and alcohol was 37%. And these are the major factors which contributes for the cause of urinary bladder cancer. But in the case of healthy control was 9 and 8 respectively. The food habits was similar in cancer patient and healthy controls. Like 39% was non-vegetarian. In the case of healthy controls was also 39%. Let's come to the result part. So this is the figure 2, which represents the heavy metal concentration. This is the figure 2, which represents the heavy metal concentration present in the blood. We have quantified 5 different types of heavy metal present in the blood. Chromium, nickel, arsenic, cadmium, lead. This is a kind of trace metal. So on the left, on the y-axis, it is representing the concentration of heavy metal in the part per billion format, part per billion unit format. And we found in all these cases, there is a differences between the control and the cancer patient, healthy control and cancer patient. But statistical significance we have found only in the lead concentration in which are present in the blood. We have used here a box plot, minimum to maximum range, for to show the because our data was not in normal distribution. So here we have used the box plot for to show the median and interquartile range in the control, healthy control and cancer patients. And out of 5, we have found the significance data with the p-value 0.02 in the case of lead in blood only. Out of 5, we have found the lead concentration in blood, which is significantly different in healthy control and case between the healthy control and cancer patient. And rest 4 sample, rest 4 heavy metal is not showing any kinds of significant difference. Here is the clinical difference are present, but there is no significant difference, statistically significant difference. Now, this figure 3 is showing the concentration of heavy metal present in the urine. Here again, we have shown that here again we can see that our data is representing in the form of box plot minimum to maximum, because our data was not in normal distribution. So here we have used the box plot for to show the median and interquartile range, and for to show the differences between the control, healthy control and the cancer patient. So on the y-axis, it is representing the heavy metal concentration, which is present in the part per billion unit format. There is the significant difference in the case of chromium between the healthy control and cancer patient. In the case of nickel, there was not so significant between the healthy control and cancer patient, because the p-value is not less than 0.05. P-value here is the 0.42. In the case of arsenic, there is again significance that we have found with p-value 0.0070. In the case of cadmium, we have found the significance difference with the p-value 0.013. Here in the case of lead, again we have found the significant difference between the healthy control and the cancer patient, where the p-value is 0.0034. Here, because of some outliers, the median value is higher in the case of control, healthy control compared to the cancer patient because of some outliers. So after removing up some outliers, we can find that difference between the healthy control and cancer patient. In figure 4, it is representing the comparison between the cancerous tissue and adjacent healthy tissue based on the percentage of 5-methyl cytosine. On the left axis, it is representing the 5-methyl cytosine percentage. On y-axis, it is adjacent tissue and cancerous tissue, where we have found a significant difference between the adjacent tissue and cancerous tissue based on this 5-methyl cytosine percentage, based on the p-value 0.0313. So there is the significant difference between the adjacent tissue and cancerous tissue based on the 5-methyl cytosine percentage. This figure 5 is representing the correlation between the heavy metals concentration which are present in the tissue, cancerous tissue, and 5-methyl cytosine percentage which are present in the cancerous tissue. So correlation ranges from -1 to 0. If the value of R is the correlation coefficient. If R value is close to -1, then we can say that the correlation between the two groups is negative correlation. If R value is 0, then there is the no correlation between the two groups. If R value is close to +1, then we can say that there is the strong positive correlation between the two groups. So here, based on the R value, we can decide that what kind of correlation are present between the two groups, like in heavy metal concentration and 5-methyl cytosine percentage. So here we have found that in the case of chromium, there is the weak correlation because R value is 0.1166 with a p value 0.41. There is not so statistically significant, and neither is statistically significant and nor strong correlation. There is the weak correlation because R value is 0.11. But in the case of nickel, here we have found the significance correlation and R value is 0.28. So here we can say that there is a strong correlation compared to this chromium because R value was 0.2834 and p value was 0.046. In the case of arsenic metal and 5-methyl cytosine percentage, there is again significant. There is not again significant. There is the little bit strong correlation comparison to this compared to this chromium metal with the p value with the R value 0.19 and the p value 0.17. There is no significant correlation. In the case of cadmium, there is a strong correlation between the heavy metal concentration cadmium and 5-methyl cytosine percentage with R value 0.4084. So here is the strong correlation compared to all these type of correlation, all these heavy metals. This cadmium metal is having strong correlation compared to all these metals with the significant correlation with p value 0.0032. Whereas in the case of lead, the R value was 0.4. Here again is a significant correlation with the R value 0.40 and p value 0.0033. This D represents the 5-methyl cytosine percentage in the adjacent and 5-methyl cytosine percentage present in the cancer tissue. There is the again positive correlation and significance, statistically significant. So based on the above result, we can conclude that heavy metals may be one of the major risk factors we cannot say exactly that he is the solely responsible for the cause of this urinary bladder cancer. These heavy metals can be or may be one of the major risk factors for the development or for the cause of this urinary bladder cancer by induction or by inducing the epigenetic modification as DNA methylation. And also it is showing a positive correlation and statistical significance between the heavy metals and DNA methylation. So our future studies can be future studies whole genome bisulfite sequencing. By using this whole genome bisulfite sequencing, we can elucidate the gene specific methylation which were caused by the heavy metal toxicity in urinary bladder cancer. These are my references which I have used in which I have used for this presentation. And this research project was funded by ICMR, that is Indian Council of Medical Research. Thank you so much. Thank you for listening patiently.